in my lab. He told me yesterday that he graduated undergraduate 10 years ago this month. And I thought, wow, that's pretty impressive. Well, what he did, that was from the University of Minnesota. After that, he went to prevention genetics, where he worked for 6 years as a, what was your title?
R&D tech too.
I was going to say technician, but
Yeah,
I wanted more specific, R&D
no,
technician.
it's
But we were really fortunate for that he decided to go back to graduate school and he came here to the University of Wisconsin. He joined my lab in 2021, and he's really taken on a complex project that involves looking at whole genome DNA methylation sequencing. And his interest is really trying to use that information to understand the dichotomy between sexes in terms of Alzheimer's disease. So I'll let Phil go ahead and tell you all about
okay. Thank you, Reed. Okay, so I guess we'll just hop right in. So I guess my thesis work is looking at blood DNA methylation changes and mild cognitive impairment in Alzheimer's disease. So just starting with a little bit of background, I don't need to go into too much detail here. We're all probably aware Alzheimer's disease is a growing problem in the US and throughout the world, with cases expected to double by the year 2050 to over 12 million within the US. And this is leading to astronomical healthcare costs associated with care. And so about 10 to 18% of adults over the age of 60 will develop mild cognitive impairment, which are declines in memory, language, and executive functions. But importantly, these individuals are still able to perform everyday of life tasks independently on their own. But 80% of these individuals within 6 years will develop Alzheimer's disease. So Alzheimer's disease is characterized by progressive neurodegeneration leading to loss of neurological function. Symptoms progress all the way to loss of independence. It affects 1 in 9 adults over the age of 65, with 2/3 of those individuals being females. And the exact mechanisms behind Alzheimer's disease are still not known, but several molecular etiologies have been identified. So that includes tau neurofibrillary tangles, beta amyloid plaque formation, and neurodegeneration. And it's important to kind of pause right here and kind of point out that there's kind of two different forms of Alzheimer's disease. You have early onset Alzheimer's disease, which only accounts for about 10% of cases, but has a clear genetic basis with mutations in PCN1, PCN2, or APP. And then there's late onset Alzheimer's disease, which is what we're interested in, and it accounts for the vast majority of cases, but has an unresolved genetic basis. But there have been several risk factors that have been identified, such the best known being the ApoE4 genotype, but several others have also been identified. There was also a recent GWAS study in 2022, which identified 75 loci associated with AD when looking at almost 800,000 participants. And so based on these studies, the estimated heritability of AD is about 60 to 80%. And so obviously that means Mendelian genetics does not fully explain AD incidence. So our question for our lab is kind of, does the environment influence the development and progression of AD? And so the best way to kind of study this is obviously a monozygotic twin study. So this first study identified 55 monozygotic twin pairs that were discordant for Alzheimer's disease. This next study then kind of took next step forward and looked in brain tissue and identified differences in global DNA demethylation in AD in the discordant twin pairs. And lastly, another study even expanded this further out to peripheral tissues and identified differential methylation in blood in discordant monozygotic twin pairs for Alzheimer's disease as well. And so I guess kind of just a quick refresher for everyone. We study DNA methylation in our lab. And so this is a covalent modification onto cytosine, typically in a cytosine guanine or CpG dinucleotide. This modification is modifiable and influenced by the environment, and it's variable throughout your life. And a key aspect is it alters gene expression. And so in this kind of just simple pictorial diagram, you have an unmethylated promoter, which leads to a transcription factor binding and allowing gene transcription. Whereas when that promoter is methylated, the transcription factor can no longer bind and you don't get any gene transcription. So how do we detect DNA methylation? The vast majority of studies to date, including all AD related studies have used DNA methylation arrays, which look at respectively less than 4% of the entire human genome. And so what we're using is whole genome methylation sequencing, which gives us coverage of over 22 million CpG sites throughout the entire genome. So our lab is focused on investigating easily accessible tissues such as blood to identify episignatures of disease. So we can take a cohort of study participants, collect blood, do DNA extraction and whole genome methylation sequencing, and then ask, can we actually separate these individuals out based on episignatures? And this is to improve detection and prognosis, improve our understanding of individual pathologies, as well as identify novel therapeutic targets. And so if it's not clear already, we do work with human data and human participants in our lab. And so kind of just a little RCR on human subjects research. So kind of just a couple highlights. So we have an IRB approved protocols. Kind of high points of that are like making sure all of the research participants had informed consent. For us on the researcher side, we have to have our appropriate training such as CITI and HIPAA. Data access has to be in a controlled manner. So we only have one collaborator who has access to participant medical records. Anything that we use, the participants have been de-identified. And then we also limit the amount of protected health information that we also utilize for our analyses. Data storage has to be in a HIPAA compliant controlled access. So for us, that is using Silo, our own departmental server, SMPH S3, and Globus. And this is kind of a big problem because as I'll show you with this project, we have over 50 terabytes of just raw data for this project, and that's not including any type of redundancies. So this is a big deal kind of to keep in mind when we're doing this analyses. And then lastly, public access, again, has to be in a controlled manner. So that there are sources such as dbGaP or the Alzheimer's disease sequencing project where we can deposit this data. Okay, so our study demographics. So we have 380 participants that were from the Wisconsin Registry for Alzheimer's Prevention or the Wisconsin Alzheimer's Disease Research Center. So we have 173 cognitively unimpaired individuals, or CU, 99 with MCI or mild cognitive impairment, and 108 with Alzheimer's disease. And it's just important to note that all these samples are also roughly sex matched as well as matched for age and education. And so we generate whole genome methylation sequencing data for all 380 of these participants. And last summer, I showed you guys the group comparison. So comparing AD versus CU participants. I then moved on to sex-specific comparisons. So looking at females with AD compared to females that were cognitively impaired. And so we left off basically needing to do the sex chromosome analysis, which was going to be a key point in my thesis. And so with this, when I started actually doing this analysis, I kind of started noticing a few irregularities in our output data. And so this includes, we had 2 females that were missing chromosome X count, methylation count files, as well as 5 males that were missing chromosome Y count files. And so we ended up just kind of taking the easiest approach, was like, let's just try realigning these samples and see what happens. And we confirmed with that realignment that we did in fact have coverage on X and Y for all 7 of these samples. But not only did we have coverage, the coverage was actually significantly better than all of the other samples that we had at the time. And so at that point, we made the decision to actually realign all of our samples from this data set. And so this is just kind of a violin plot looking at the number of missing values on chromosome X from the 380 participants. So we can see here there's 1.3 million CpG sites on chromosome X. And so basically, the vast majority of samples are missing around just over 150,000 sites, I believe, compared to, but we also have samples that are outliers as well, missing over 90% of the CpG sites on chromosome X. After realignment, all samples now have less than 50,000 missing sites. And so this kind of led us down a little troubleshooting route that I kind of undertook. And so I don't have time to kind of go into all of the details of everything we looked at, but kind of just a brief overview of how this data is analyzed. We start out with raw sequencing reads. These get trimmed for low quality bases as well as adapter sequences. We then align them to the reference genome using a methylation aware aligner. And then we can actually do the methylation calling at that point. So that's actually looking at each cytosine and identifying how many reads are methylated and how many are unmethylated. And then from that file, we can actually do our downstream differential methylation analysis. And so with the troubleshooting, I was actually able to determine that we actually had at least two problems at both the alignment and calling steps. So at the alignment step, if we compare the old and new alignment files, we know that there are differences between those. But we also know at the calling step that there were issues as well. So there was only a modest correlation between the new output calls, as well as obviously we were missing these call files in those 7 individuals from the get-go. Okay. And so with all of this happening, we also decided to kind of do one additional thing that to check. And so this was actually screening for early onset Alzheimer's disease within our samples. So obviously the goal here is to screen for variants in PSEN1, PSEN2, and APP. And the reason we're doing this is because, to our knowledge, this is the first time any participants from RAP or the ADRC have been sequenced. And so we weren't really sure if these could have come through. And so essentially what I did was I kind of just generated my targeted regions of interest. So that's using the main select transcript for APP, PSEN1, and PSEN2. I took out all of the coding regions and added 2 base pairs of flank onto each exon, so that way we can include canonical splice donor and acceptor sites, and then filter out all the variant call files for all 380 participants, and then annotate all of those variant calls with ClinVar annotations. And so here's just an example of a mutation. So this is in PSEN2. This is an adenine to thymine mutation, so right in the center. And so you see the reference A. And basically, you can see about roughly 50% of the reads are also showing T. It's important to note here as well that you can see neighboring are all of the converted cytosines, which is how we detect methylation as well. So again, you have to use a methylation aware variant caller to actually be able to do these variant calls. So findings. So to our surprise, we actually found 4 individuals with pathogenic variants in PSEN1 or PSEN2. So these would all be considered early onset disease, early onset Alzheimer's disease participants. So obviously at this point, we decided to remove all 4 of these participants from the study. But additionally, we also found another 11 variants. So 6 that have been reported as or 5 that have been reported as uncertain in ClinVar, as well as 6 more that have not been reported anywhere. 10 of them are missense, 1 nonsense mutation in APP. And all of these variants are very rare comparatively. And so without actually knowing kind of what these variants are doing, we actually opted to remove all these additional 11 participants from our study as well. Okay, so now that we've kind of cleaned up the data and we removed a few participants, we can kind of now hop back and look at some of the sex-specific DNA methylation differences between AD, MCI, and CU and blood that I presented last summer. So just as a reminder, we can kind of think of this in pairwise comparisons. So kind of looking at AD versus CU, are there DNA methylation changes associated with AD? MCI versus CU, the same thing in AD versus MCI. Are there methylation differences between MCI and AD? And so when we actually do this differential modeling, I start out by filtering out CPG sites to make sure that there's at least 10x coverage in all participants. So that left just over 22 million CPG loci for analysis. We can then do a PCA on this data. And so this is kind of just a showing the PCA on the most variable CPG loci. And so interestingly here, we find that our participants or kind of separate on the principal component one by ancestry. And this is what we have observed with other whole genome methylation sequencing data sets in the past, which is giving us confidence that our newly realigned data is actually correct. And then our actual statistical model. So again, our dependent variable is going to be the actual CpG methylation. And then our testing hypothesis variable is essentially testing whether that coefficient is equal to 0, being the null hypothesis, or not equal to 0, which is our alternative. We include other covariates in our model, such as BMI, age, white blood cell estimates, and then PCAs as well. And then as what I define as the differentially methylated position is having a false discovery rate less than 0.05, and then a mean methylation difference between groups that are being compared of at least 2.5%. And so here's a volcano plot looking at the male AD versus CU comparison. So on the x-axis, again, is going to be the mean methylation difference between groups. And then the y-axis is looking at the significance. And so colored points, each point represents an individual loci. And so here we can see that I have 11,427 hypomethylated positions and then 81 hypermethylated positions in this comparison. We can take those 11,500 DMPs and actually ask how do these fall within the genome. And so interestingly, we find about over 22% actually fall within promoter regions. So that's defined within 3 kilobases of the transcription start site. And surprisingly, like only about 18% are found in distal intergenic regions. So not near a gene at all. And so if we take all of those DMPs that are within genes, that totals a total of 4,806 genes that have at least one DMP in them. And so here's kind of just a couple of gene schematics to kind of see where this differential methylation is falling within gene. So kind of just looking at this first example, kind of going from top to bottom, you have a scale bar at the top followed by HG38 genomic coordinates. And then you have the lollipop plots. So the height of each lollipop represents the significance, and each lollipop is a differentially methylated position. So white circles indicate hypomethylated positions, and then black circles indicate hypermethylated positions. Below that in blue are the gene schematic, and then below that in green are CpG islands annotations. And so I just chose 2 genes here. DLGAP2 has 11 differentially methylated positions in it, and it's a neuronal signaling and synapse organization gene that has had shown that decreased expression has been associated with AD pathology. SND1 is another gene. It's a transcriptional coactivator and can interact with APP, and it has 9 differentially methylated positions within the gene. And so then lastly, we can then ask, like, we have all of these genes with differentially methylated positions. We can actually ask, how are these genes potentially related to each other? And so we find numerous gene ontology terms, such as this vesicle-mediated transport in synapse, microtubule-based transport, and then molecular function. We have protein serine kinase activity, as well as other epigenetic regulatory items such as histone kinase activity. Again, and so we kind of then the next question we asked was basically we had those 18% of differentially methylated positions that didn't fall within a gene, and so the next question was, can these actually still have some type of functional relevance? And so what we did was we overlaid these differentially methylated positions onto a promoter capture Hi-C data set that was specific to blood. And essentially this data set links enhancer regions to promoters of genes. And so here's just another example, the same kind of setup, except now you have these arches below, and that's linking the enhancer region marked by E to various promoters upstream and downstream. And so if we kind of just as a whole We find that there are 669 enhancer regions that have at least a differentially methylated position in them. And these interact with at least over 1,000 gene promoters throughout the genome. And if you kind of like ask what those genes were kind of going or related to, most were related to cytoskeletal organization as well. And then another thing that we could look at also is motif enrichment. So are these differentially methylated positions potentially interacting with transcription factor or DNA binding proteins? And so essentially what I did was I took each differentially methylated position, added 25 base pairs of flank onto each side, and then used Homer to actually identify enriched motifs in our data set. I'm showing the top 9 with a false discovery rate less than 0.01. And I'll just point out, second from the bottom, the LXRE is a liver X receptor elements, which regulate cholesterol metabolism, ApoE expression, and our potential therapeutic target already identified for Alzheimer's disease. So that was really interesting to see when we got this data back. Okay, so that was all for males. I think so now we can actually do the same for females. And here we only find 9 differentially methylated positions, all hypomethylated, when doing the female AD versus CU comparison. These 9 positions annotate to a total of 7 genes. And then the 2 genes that are asterisk have 8 AD relevance with them. We can also then obviously expand this to the other comparisons. So if we look at MCI versus CU, we find 1 differentially methylated position in both males and females. And then if we also look at AD versus MCI, we only find 1 hypomethylated position in males. Okay, so kind of just in summary of these sex-specific comparisons, we find differential DNA methylation in blood is limited to autosomes when comparing AD and CU participants in males. These majority of these DMPs fall within or near gene bodies. DMPs were found, for example, in DLGAP2, who has had AD-related evidence. And then gene ontology results also highlight processes involved in neuronal signaling and connectivity. Again, intergenic DMPs were located in enhancer regions, potentially affecting gene expression. So again, FLOT2 was one of those promoters that's interacting with a differentially methylated enhancer, and it's involved with vesicular trafficking, signal transduction, and APP endocytosis. And then lastly, differentially methylated positions were enriched in multiple transcription factor binding motifs. And again, the liver X receptor elements have already been targeted as a potential therapeutic target. Okay, so moving on to the sex chromosomes, just a refresher that these are typically excluded from EWAS studies. So this is mainly because of chromosome copy number differences as well as X chromosome inactivation. So what we have to do here to actually analyze is we actually have to sex stratify our cohort to do this analysis. And so again, we have the same model, except in this case, we're just looking at the cognitive status variable since our data set is already sex stratified. And then we use the exact same covariates as well. And so here, I guess, essentially we find no differentially methylated positions in all 3 of these comparisons. I know I'm kind of running short on time, so I'll kind of just leave you with a couple pieces. So one, we kind of think we might be running into a power issue with these since we're essentially using only half of our samples now to detect these differences. And then there's also the possibility that the sex chromosomes aren't, or at least certain sex chromosomes are not actually that important. So I guess one highlight would be the male chromosome Y. With age, actually in blood, males actually, there's a thing called a mosaic chromosome Y loss. And so you actually lose your Y chromosome over time with age, which we actually can show with our data as well. So lastly, I just kind of want to talk about what I'm really focused on right now, which is analyzing methylation quantitative trait loci with the STAT or methyl QTLs. So The goal of this portion of my project is actually to identify genetic variants which are correlated with changes in DNA methylation at specific sites. So in this simple pictorial diagram, you have an A allele, which is correlated with downstream methylation. When you have a G allele, that correlation goes away, or that the methylation goes away. And so how can these like come about? So like in its simplest form, you have a CpG SNP. So you normally have a methylated cytosine. You have a mutation, so there's no longer a cytosine there. It can't be methylated. That's your simplest methyl QTL. But you can kind of think more locally, where you have a transcription factor that binds, that promotes demethylation of an area. But when you have a mutation, that transcription factor can't bind anymore. That region is now can become methylated. And then you can think kind of more globally, kind of a more trans mechanism where you have that cis-methyl QTL, which affects the expression of an epiregulatory gene, and that now causes widespread genomic changes. And so I kind of just have a preliminary example that I just want to show everyone. So I took one risk SNP, so that's in the gene CR1. I have the 3 genotypes shown, so 250 homozygous alternate, 111 heterozygous individuals, as well as 19 homozygous reference individuals. And so you then compare that to all CpGs just on chromosome 1. So that's just under 1.9 million cytosines. And so we identified 15 methyl QTLs with an FDR less than 10 to the negative 10th power. And the top methyl QTL was about 48 kilobases upstream within another intron of the gene CR1. And so this is kind of just a plot now looking at the actual methylation differences. So on the x-axis, we have the 3 genotypes, and then the y-axis is the methylation at that upstream CpG site. So when you have the CC allele, generally speaking, almost all the samples are very low or almost 0% methylated. When you're heterozygous, it's in the 50 to 60%, and then homozygous reference or TT, you almost have a fully methylated position. So again, just kind of proof of concept, but a really cool finding. And so essentially, next steps for me with this analysis is obviously expanding this out to all 75 AD risk SNPs, as well as all CpGs throughout the entire genome. I'll be doing additional filtering and quality control. So obviously we need to have all genotypes represented when we do this analysis. So there are some variants that are going to be too rare for us to actually analyze. But there are plenty that we can. And then we also need to just double check Hardy-Weinberg equilibrium is okay. We can further refine the model, so add in additional covariates, as well as adjust our cis and trans parameters. And then from there, we can just do downstream analyses such as clustering these into differentially methylated regions, as well as annotation. And so with that, I just want to thank our lab, and then a few of our collaborators, kind of just a shout out to Lindsay Clark, Kirk Hogan, and then a former lab member, Dr. Andy Madrid, for their help on this project. And then I know we're kind of short on time, but I can take any questions. Oh, or she?
So, in blood samples for cleansing,
Yep.
and DNA methylation is relative to context specific. So if you did not want to know, I understand that you might not have access to brain samples, but do you happen to know how well it correlates?
It's definitely like we've done mouse studies and it's in the like 70% range. We've seen we are getting some brain samples, unfortunately not from the same individuals, but yeah, it's definitely a question for sure. We at least attempt to control as much as we can with this blood cell type. When we model, we include the different the the individual white blood cell types that way to help better account for the methylation differences.
And that you can maybe use to filter out
I don't know if we've ever actually looked that close at which ones didn't correlate that well. But that's definitely a good thought to do in the future. Manny.
Um, yeah, I was thinking about like the age of the patients of your blood samples, and I was wondering if there's something that you're doing in your analysis pipeline to account for just aging related methylation.
Yeah.
Because
Yeah. So we do include participant age in our model. The blood samples are also all from essentially their closest visit to their diagnosis. So we hope to at least it's trying to be as much of a snapshot as we can. So kind of within a few years of everyone being diagnosed, it's the most that the same blood sample essentially, even though it's happening longitudinally over time. Yeah. Dr. Gash.
I'm curious about the small subset of samples that had pathogenic variants in the early onset.
Yep. Mhm.
I guess they must have gotten Alzheimer's to be in the study. Do you think that so what's the explanation? Is their onset of disease related in the same time frame as early
Yeah.
onset, and they just kind of run the.
Yeah, so the whole timing wise, I can tell you, so the two youngest Alzheimer's disease samples were two of those people with pathogenic variants. So that made sense. But yeah, the whole it used to kind of be a hard cutoff 65 or older. And that is kind of going away at this point. It's definitely broadening out. Um, Overall, most of these, even some of the missense variants as well, were on the younger side as well. And so that's kind of was more evidence that we probably should just exclude all of these participants.
Just a misstep.
Yes. Yeah, and as I said, we also, to our knowledge, these are the first time any of these samples are being sequenced. So we've actually notified the RAP and ADRC directors that hey, these samples exist, and kind of pushing them to you should maybe at least start considering doing some sequencing on these individuals in the study as well. The only thing that they have done currently is just an ApoE genotyping assay. But other than that, to our knowledge, there's otherwise no sequencing. We do not. So these are all obviously they were collected with the rapid ADRC, and so it's just biobanked blood samples, and so that's been frozen. So as soon as we thought it's all white blood cells, and so you just released all of the RNases into essentially the sample, and so we've started exploring a few ways of if we could possibly try to. There are some methods that have limited success at kind of holding the RNases at bay. But as of now, we don't have RNA expression data sets. There are publicly accessible, but it's not matched with our data. Yep. Yeah, for sure.